Good morning, everyone. We're waiting for guests to trickle in, and I'm going to share my screen. I have a few introductory slides. Here we go. I'm going to start in 1 minute. Okay, I'm going to get started and go through the introductory slides. So welcome to the wastewater surveillance for SARS-CoV-2 and emerging health threats continuing webinar series. This is a continuation of the RCN webinars. If you don't already have these links, we have a mailing list registration and also a speaker nomination. If you want to take a picture of either one of these, you can nominate a speaker for the webinar or register for the mail list if you're not already on it. Today we have two speakers that I'm very excited to hear from. One is Dr. Sheena Conforti, who's a postdoc at Stanford, who's going to be talking about enterovirus D68, and then Miranda de Graaf, who's an associate professor at Erasmus University Medical Center in the Netherlands, talking about the global urban virome. And the way the webinar works is there's going to be a Q&A that you can see at the bottom or top of your screen where you can put in questions. And Sarah Philo, who is a postdoc at USC, and I will be moderating the questions for the speakers. So we'll remind you as we go along to put questions into the Q&A. Okay, so with that, I'm going to hand it over to Sheena, who can share her slides and begin.
Thank you, Ali. Good morning, good afternoon, everyone. Thanks a lot for inviting me. I'm Sheena Conforti, and I'm a postdoc, as Ali said. And when I arrived at Stanford some months ago, I had this huge amount of enterovirus D68 concentration data in wastewater all around the US. And I actually was wondering, what can we do with this huge amount of data besides seeing if they correlate with clinical cases? Can we do answer some epidemiological questions about the circulation of this virus across the country? And can we see if the circulation pattern do depend on some climatic factors or social demographic factors? And so, for who doesn't know it, enterovirus D68, it's a respiratory virus. It can cause mild upper respiratory illnesses such as cough, cold, and so many adults, people especially, they are not diagnosed when they are infected with EVD68 just because it causes mild symptoms. But then sometimes, especially in children and elderly people, but not only, it can also cause lower respiratory illnesses such as hypoxia or wheezing. And unfortunately, especially in children below 5, but not only, EVD68 has been associated with complication to the central nervous system and can cause subsequent diseases such as the acute flaccid myelitis, which is partial paralysis in children especially. And so it has been shown that kids that were actually infected with EVD68 1 or 2 months later might develop this syndrome. And so Unfortunately, there is no vaccine available for EVD68, and there is sparse clinical testing. There is no standardized laboratory procedure to test for EVD68. So, for example, in the US, we know that there is a certain panel that tests for RSV, influenza, SARS-CoV-2, and EVD68 is not part of it. And what we know about EVD68 from the past is that it is not like the other respiratory viruses, but it has a biennial outbreak pattern. So it circulates usually only every 2 years. Till now, it was mainly even years, so 2014, 16, 18, and so on. And it outbreaks mainly towards the end of the summer and the beginning of the fall. So it's considered like a summer virus. From previous studies, we measured actually EVD68 in wastewater in the US, and these previous studies of Ali show actually that the concentration of EVD68 measured with dPCR here on the top two graphs increases in at the end of the summer in the fall of 2022, and this increase in concentration correlates with laboratory confirmed cases in California. So we do already know that what we measure in wastewater actually matches what is circulating in the clinic with infections numbers. Now, what I wanted to do was like, can we use wastewater data only to try to infer when EVD68 starts circulating and for how long it circulates, both at national scale and also at local scales, for example, state or wastewater treatment plant. And to do that, using wastewater data only, so concentration measured with digital PCR, I tried to estimate the so-called center of season, and it's actually the peak, and this is this vertical line here, which doesn't necessarily is the highest concentration measured, but it accounts for the whole season. So let's say we have like here two small peaks, then the center of season is in between these two, and the season duration. So for how long can we detect EVD68 in a specific location? And so I use almost 44,000 wastewater samples analyzed between July 2023 and July 2025. We measured EVD68 concentration from 147 wastewater treatment plants all across the US, specifically 40 states. You can see here the black dots on this map. And we detected this virus in 24% of wastewater samples. It seems low, but if you consider that actually is a biennial outbreak pattern pathogen, then it's more than understandable. And so, aggregating all this concentration around the country, we actually observed that, as expected, the center of season fell in September 2024. So we have almost no detection in 2023, and then it decreases on the following year. However, the season duration is pretty long when we aggregate the concentration at national scale from all these 147 wastewater treatment plants, and we see that it starts in February 2024 and it lasts until the following year, May 2025. And here you can see that actually, especially in January 2025, there was a specific peak that usually is not there based on historical data that we have about EVD68. And this is actually due to a specific state, which is California. So if we look at California, we see that the center of season, as I said before, is not really the peak, but is shifted to account for both this peak. And it happened in November 2024. And in California, we have a usual increase towards the end of the summer, beginning of the fall 2024, but then a second peak in January 2025. And we also observed that the season duration here, the light blue shadow, actually lasts pretty long. And so it lasts until when we cut the data set to perform this study. And so it lasts more than 20 months. And this is pretty untypical because for the majority of the states, such as Pennsylvania, in reality, we observed a center of season as expected around September or October 2024, and then a shorter season duration of a few months. And so we estimated the center of season for all the states and we plotted them on this map. And we clearly see that it varies between June 2024 and November 2024. And if you look at the pattern, it starts circulating earlier in the southeast of the country, and then it moves north and west. And so we checked if this has a correlation with latitude. And as you can expect, EVD68 circulates earlier in the south compared to the north, but also circulates earlier in the east compared to the west. Now, this is very nice because we estimated this timing based on wastewater concentration only. But in the past, a study from Ponsalort et al. In 2018, they used enterovirus surveillance data collected over 30 years. So not only EVD68, but enterovirus in general. And they observed something very similar to us using this clinical surveillance data. So they have the mean timing of cases here going from 6, which is June, to 11, which is November. And we clearly see that there as well, we observe earlier outbreak in the southeast of the country compared to the north or the west. And so this shows us that using wastewater data only, we can actually estimate this circulation pattern very well. And this was only 2 years of data compared to clinical cases and over 30 years of collection data. Now, if we move to the season duration, we also observe some pattern at national level, and we see that there are differences on a spatial scale. So usually, as I said before, like for Pennsylvania, we have, for the majority of the states, this EVD68 circulates between 1, 5, 6, 7 months. But then we have a few exceptions, such as Florida, a bit longer duration in Texas, and a very, very long duration in California. And so I don't have an answer on why in California is like this, but one of the questions we might ask ourselves is EVD68 actually shifting its biennial pattern towards a more present and annual outbreak, as many other respiratory viruses already are. And here we observe as well some correlation with the latitude and the longitude, even if it was not as strong as for the center of season. Now, if we see such a map, the US are really huge. I mean, I come from Switzerland and it's very small. And we can also assume that north, south, west, east, they are characterized by different climatic factors, for example. And so the next step was to see what drives this seasonality. Is this seasonality due to climatic factors? And how does sociodemographic indicators as well might affect when the virus circulates, so the center of season, and for how long the virus circulates. And so we selected various climatic factors such as temperature, wind speed, the dew point, relative humidity, precipitation, and pressure. And we observed that the center of season highly correlates with temperature and the dew point. And the dew point is actually a measure of temperature and humidity approximated. And so we see that EVD68 circulates earlier in warmer and moister locations. And here, each dot corresponds to a different wastewater treatment plant. So instead of estimating this center of season at the state level, we did it at really scale level, so local scale level at the wastewater treatment plant. And so this is very clear. And the point is like temperature and dew point both correlate between them. So we cannot really differentiate which one of these variables explain more what we observe in the center of season variation, but they also correlate with latitude and longitude, as I said before. And so we try to see if the addition of temperature or dew point to a geographic model only might add some explanation for the variation in the center of season. And we see that latitude and longitude alone, they do explain 60% of the variation in the timing of EVD68. But if we add either temperature or dew point, then both of them almost increase this explanation of 20%. And so temperature alone might explain at least 20% of the variation observed in the center of season. Very interesting. EVD 68 timing depends on these environmental variables, but the season duration is not correlated at all with any of these environmental factors, as you can see from this plot. Differently, this season duration here plotted in weeks is actually correlated with sociodemographic factors. And so here we collected many sociodemographic factors at local scale again. So each dot corresponds to a different wastewater treatment plant. And we see, for example, that in location with a higher childcare density per square kilometer, the duration of the season of EVD 68 is much longer compared to location where this childcare facility density is lower. And this is also observed depending on the proportion of crowded households, so namely the households in which at least two people per room inhabit the household. So where there are more people living together, the duration is longer. Where there is a higher population density, the duration is longer. Where there are more nursing home, more hospital, and where there is a higher rate of urbanicity. As before, very interestingly, these sociodemographic factors do not explain any variation in the center of season, namely in when the virus actually starts circulating, except here for the childcare density. But we don't see actually that a higher number of nursing home or hospital is associated with an earlier or later center of season, nor the population density or the urbanicity. And so this brings us to actually the main conclusion of this short study, which is that environmental conditions apparently trigger the onset of EVD 68 detection, namely when it occurs, but how long it lasts in a specific location depend on demographic structure and social mixing. And so this is very nice to know. And actually, it's very nice that we could show it using wastewater data alone without relying on clinical data. And so this brings me already towards the end of the presentation, which is that a wastewater provide dense data, and it's a lot of data, especially nowadays, especially after the SARS-CoV-2 pandemic, where globally we started producing so many wastewater data, right? So we can really use them to reconstruct this circulation pattern of many viruses and pathogens, especially for which we do not have clinical data that are standardized globally or at national level. And so what we can do afterwards, it would be very interesting actually to also incorporate strain level typing to assess if there are differences in lineage specific circulation pattern. But it might also be very interesting to improve the interpretation through shedding studies. So now we know that what we measure in wastewater is we measure actually a trend of what's circulating in the population, but it might be nice to refine this link between RNA concentration in wastewater and really like the number of infected individual within a community using concentration that are shed by each individual. And another take-home message from my side is that, as I said before, warm and more humid region experience in the US earlier EVD 68 circulation, and population density and social structure actually influenced very well how long this season persisted. And so I would be very interested in to see what are the effects of climate change on EVD 68 seasonality. So how can we use this wastewater data to model future climatic condition and to try to understand how we can use only this wastewater data actually to model the circulation, future circulation of these viruses for which we do not have a lot of clinical data available. And secondly, how can we translate wastewater detection really into action? So in this example, we saw that childcare facility density really affect how long EVD68 circulates in the US. So once that we start monitoring and detecting EVD68 in wastewater, can we really do something concrete in these facilities in order to decrease its season, and how can we do that? And then what this study brought me actually to the motivation of using wastewater data behind the correlation with clinical trends towards really trying to characterize this circulation pattern and try to use this wastewater data to answer some epidemiological questions for which we do need a lot of data and for which often we don't have just because clinical surveillance is not as standardized as for other pathogens. And so, said this, I really thank you for the attention, and I'm very open to any question that you might have.
Thank you so much, Sheena, for that really interesting talk. We'll get into some questions. So, I guess I'll start with one that I have about the California data. Have you considered breaking that up into like regions of California, since the climate of California is very different depending on the north or the south? Maybe that would like affect the peak. Is that something that you've considered?
Yeah, this is a great question, and actually is one of the things we did as first. And I'm not showing it here because there was no trend.
Okay.
So we know, for example, that in San Jose, so south of the Bay Area, it just lasts for all the 80 weeks. It's really incredible. And in further in the south, there are for some counties it just lasts like a couple of months, but in the north, then again only a couple of months. And so that's why when we instead of at state level, we estimate these metrics at wastewater treatment plant level.
Mhm.
We see this nice temperature, for example, effect or a sociodemographic effect.
Okay, interesting. So looks like there's a couple questions. I encourage everyone to keep putting their questions into the Q&A. So, um, you talk about temperature as sort of triggering these outbreaks. Is there any ideas that you have about the mechanism of that? Um, like, are there any lab experiments you could do to maybe, estimate that? It's a really interesting data there.
Yeah. So this is something I might not have the expertise to answer completely, but so we know that EVD68 is a summer virus, right? They define it like this. And so that might be because warm and humid condition create some favorable microclimates for, I don't know, droplet mediated transmission. And so they maintain the viral stability and they prolong the survival of these infectious particles in the air and especially on our mucosal surfaces. So this might be one of the reasons. I don't know if I completely answered the question, but we don't know. And so some experiments might, I don't know, might be try to see how this virus survive under a different temperature and under different humidity condition, I would say.
Yeah. Seems that there's a lot of open questions about the specific epidemiology. We have a question in the Q&A about what drives the every other year cycle. I was wondering that as well. Most things are annual. If you have any idea, if there's any research or information about what that 2-year cycle is representative of.
No, I don't know. I'm not sure. I would guess it's just because It might take longer to actually become infectious again. I don't know. We know, for example, for RSV, there is A and B, and we know that one year A is dominant and the other years B is dominant, and there as well, there is a lot of uncertainty. What I find it very interesting here is that I'm not showing it here, but 2025 shouldn't have any detection based on historical data, and we did have some detection in wastewater in some states, lower than what observed in 2024, but still there. And as I showed also for California, it's always there. So my question is it actually gonna be still every 2 year, or is it gonna be more permanent and regularly there every year as other respiratory viruses?
Mhm. Yeah, that could maybe there's like an immunity question there too. Like, does immunity wane every 2 years type thing? That'd be a lot of interesting questions there. Um, I was also wondering if you actually did compare this to any sort of clinical data, like the limited clinical data that we have on EVD 68, if that work was finished at all.
So no, unfortunately, there was a shutdown, so they were unavailable to obtain, as you might know. But what we did, and I didn't show it here, but I have a slide. It's using Epicosmos data for diagnosis, and so we use, for example, here on panel C, diagnosis for enteroviruses. So when clinicians just write in this health record, okay, they have probably enteroviruses because of the symptoms they reported, and we see in blue here the number of cases of enteroviruses diagnosed all over the country, all over the period. And in black, we have the wastewater concentration. And we don't really see a huge correlation just because these proxies that we used as clinical confirmed data are not really as precise as laboratory confirmed data. So we also used wheezing, but wheezing is very general. And so we didn't really observe this correlation with wastewater concentration. If we look at acute flaccid myelitis, which is known to, they already show that actually is correlated with EVD68 cases. We can see that in few states, there kind of is a correlation, but it's really, really only in few states.
Yeah, lots of interesting clinical questions there. We have time for probably one more question. Um, if we're not seeing any more in the Q&A, does that 2 year like even year pattern hold across the world, or is that specific to like the northern hemisphere versus the southern hemisphere? Like with the flu, it rotates around the world. Um, is that 2 year even year pattern
Yeah. So,
across the globe?
Yeah, so from my knowledge, it's a 2 year even in Europe, for example, and it has had a big outbreak in 2014, if I'm not wrong, with consequences of acute flaccid myelitis as well. That's when especially VD68 got the attention of many clinicians and epidemiologists. And since then, yes, it is every 2 year, even if a previous study showed that in California, for example, in 2016, there was a very, very strong and long outbreak, followed in 2018 by a much shorter outbreak as well. So I'm curious to see if maybe this year in California will be much less and would resemble what already has been observed in the past.
Yeah, it'll be interesting to see if it starts around, February, how that data then moving forward, if these hypotheses and trends still hold, that will be really interesting to see. I think, Ali, should we move on to
Yeah.
Miranda?
Thank you, Sheena, for that
Thank
great
you.
talk. Miranda, we'll have you share your screen, and you can go ahead and get started.
So thank you for the invitation. So indeed, my name is Miranda de Graaf, and I work at the Department of Bioscience of Erasmus MC University Medical Center in Rotterdam, the Netherlands. And I don't have to convince you, but wastewater is a really nice way to survey the population. So we are very interested in it because if you think about a new variant or a new virus, you usually only notice it when people get severely ill and when they are diagnosed. Whereas whereas it's much more likely that a larger part of the population has mild or no clinical symptoms. And these people you can also monitor through wastewater. And then, of course, you can detect the genetic material, but you can also sequence. And especially next generation sequencing methods have been used more and more. And there's a couple of different strategies that you can use. So one is metabarcoding, and here you target a small region of the genomes of the typing region. And it's quite sensitive, but you do have to know what you're looking for. You can also use full genome PCRs like amplicon-based sequencing with the tiled amplicon approach, which we have used quite a lot for coronaviruses. And this is also a quite sensitive method, but you have to know what you're looking for. And especially metagenomics has the opportunity to sequence everything in a sample. So you can do direct sequencing where you essentially sequence everything. But you can also use a capture-based method where you use probes that enrich for the virus of interest. So we actually used both, so shotgun sequencing and a capture-based approach. And for that, we first developed our own probes, and we developed them based on viruses that shed through the enteric tract. And so what you can see on the right is a principal component analysis where we plotted wastewater samples sequenced with a non-capture method and with the capture method. And what we saw was that you can really enrich for viruses of interest. So of course, sequencing is one thing, but the analysis of the data is also a maybe even bigger part of the study. So you can do read-based assignments, but we chose to first form contigs. So we, of course, do the quality control and read the duplication, filter out human reads. And then we performed assemblies and annotation. And of course, you also have to submit your data. I think what is very important if you perform analysis like that, especially if you want to do typing below the species level, you really can't trust the reference databases because they're not really built for that. So we use either typing tools like those available through the RCN, or we build our own custom workflows. So the study that I will present today is the Global Urban Wastewater Study. So this was work performed together with Frank Müller-Aastrup from the Danish Technical University. And what we did is we sampled in the period 2017 to 2019, and then 52 countries and 63 cities. And in the blue dots, you see where we have biannual samples, so 2 to 4 samples in this period. And in the red dots, you can see the longitudinal sampling, so where we have much more samples. And then what we did is we performed shotgun sequencing and also capture-based sequencing for viruses. And Frank actually performed sequencing for the bacteriome. So here you can see the results. So in the top, you can see the results for the metagenomic data set, and in the bottom, the results for the gastrocap data set, and then ordered based on continents. And on the right, you can see the different hosts that the viruses infect. See if I can. Yes. So in total, we detected 122 virus families, more than 1,000 genera, and 2,500 species. What we also observed was both for the shotgun sequencing and for the gastrocap, is that a large fraction of the virome consists of bacteriophages and plant viruses. But what we also saw was that when we use this gastrocap approach, we're really able to enrich for viruses of interest, or our interest at least. Also, when we zoom in, you can see that on the left, again, the shotgun sequencing, on the right, the capture-based sequencing, that you, for specific viruses, really enrich your data. And this, by the way, was all work done by my PhD student, Natalie Worp. And she detected, of the 8 virus families that we targeted by GastroCup, she detected 72 genera and 165 different species. So then came the task to compare the virome between locations. So we performed principal component analysis. And what I've shown here is in the top, the metagenomic data, and in the bottom, the GastroCup data. And then on the left, differences based on family, and on the right, the genera. And so if 2 samples or 2 dots are very close to each other, that means that the viral composition is very similar. And if they're far apart, it's very dissimilar. And the arrows, for example, here, this arrow means that the differential placements of these samples is for a large part due to this specific virus family. And what we actually observed is when we overlaid this data is that the overall composition, if we compare it to different continents, is very similar. And then if we looked at variance between the continents, it was primarily driven by genera like the Bamovirus and Mamastrovirus. However, when we then zoomed in a little bit, so this is the data from the longitudinal sampled cities shown here. We could see that when we again plot for family and genus or genera, we can see that the samples are much more apart, and you can see differential clustering for the sampled cities. So really showing that, at least at the city level, there are bigger differences in the virome. And so also for the differential placement, for example, here you can see that Melbourne is really separate, and one of the viruses that is important for this difference is Mamastrovirus. But also enteroviruses and noroviruses had quite a big impact on where the placement was. Oh. Yes. So what we did next was we investigated several viruses that we found differentially present in the samples, and one of them was Bamovirus. So Bamovirus is a plant virus that infects several of the plants that we eat. And so the way we show it now is per continent, and then the samples are ordered based on composition. And what we found is, for example, varying levels of pepper mouth mottle virus, which is often used to as an indicator virus as well. And what we also saw was pathogenic viruses, like the emerging tomato brown rugose fruit virus. And those are indicated here. So this virus is a virus that infects tomatoes, as shown in the corner. And this is really a very pathogenic virus. So it can reduce crop yields up to 70%. It leads to mandatory crop destruction, but also import bans. So it's really traced and reported. And what we saw was that we were actually able to detect it in wastewater before it was reported in the countries shown here. And showing that next to human viruses and animal viruses, wastewater can also be used for plant viruses. So another virus that we investigated were enteroviruses, and these typically cause respiratory or gastrointestinal disease. And several of the enterovirus types are associated with severe disease, including acute flaccid paralysis, as the previous presenter also showed. And of course, poliovirus, which is also an enterovirus, was actually one of the first viruses investigated in wastewater. So we detected many different types, and I will walk you through this slide. So again, we ordered the data based on continent and also ordered based on composition. In the top panel, you can see the different species that we detected. and then below the different types that we detected. And also for this, we used a typing tool. And what we saw is that the composition differed per continent. So for example, in Africa, we found a different composition than for Europe or North America. We also found, for example, high levels of echovirus 30 in Europe, and this also coincided with large outbreaks at the time. And what we also clearly showed is that when you have more longitudinal data, it becomes a bit easier to see different patterns. So here again are the 8 different cities where we have a bit more data. Again, the different species and types, but this time it's just ordered in time. And what you now can very clearly see is that there's really quite big differences between cities. For example, Quito, Yaoundé, and Canada, you really see differences in compositions. We also observe the shifts that are we also observe shifts. For example, Coxsackie virus A6 is shown here in the earlier period in 2017, and then later you see a shift to Coxsackie virus from Coxsackie virus A10 to A6. And I have to say that this data is really based on the official typing region, so that data is a bit more limited. If you do typing based on the whole genome and also small parts outside of the typing region, you can see these shifts even more clearly. So what we also did is we investigated if we can use these sequences from Waster to build phylogenies. So we took the high quality context and built phylogenetic trees. And what you see here is all the different contexts from wastewater in circles and context and sequences from GenBank in squares. So this is mostly clinical data. And what we very nicely saw is that our wastewater samples cluster together with human samples, and often also from the same period and the same locations. So we investigated a couple of more viruses, and one of the examples that I want to show you here today is that of norovirus, because this is one of the viruses that my group works most with. It's a major cause of non-bacterial gastroenteritis. It has an enormous economic burden, and it's responsible for almost 700 million cases each year. And what is important to know for this presentation is that there's an enormous diversity of norovirus. So you have almost 10 different genogroups, and then humans are mostly infected with genogroup 2 and genogroup 1, which are further subdivided in genotypes. So we investigated if we could use wastewater also to monitor these viruses, and this was work done by Rai Scredelara, who's also a PhD student in my lab, and together with Natalie Warp, and he found more than 7,000 norovirus contexts. We detected 34 capsid and 44 polymerase types, and the most prevalent were the G13, G22, and G26. And then he next evaluated how it compared between the different continents. So what he did is one line represents one city, and then here are different continents, and on top you can see all the different genotypes. And what we saw is that some genotypes are circulating at higher levels than others, also different than suggested by clinical surveillance. And essentially, some of these genotypes you find everywhere, whereas other genotypes are very location specific. So for example, G9, we mostly detect this in Africa and not on the other continents. And for example, G227, if we look at the clinical data, it has only been reported in South America, and this is also exactly the only place where we detect it in wastewater. And there are a few more of these examples. So it also allowed us to look at shifts in composition. So what you can see here is again the longitudinal data, so the 8 different cities, and then on the left the polymerase types, and on the right, the capsid types. And what you can really nicely see if you have good density data is these shifts in composition, but also that, again, some cities are very different in composition than others. So, for example, Quito has, again, a very different composition compared to Yaoundé, but also Seattle. It also shows you that it's good to have many samples because usually not all samples will give high quality data. And I just want to emphasize that for these types of profiles, you normally would need to sequence hundreds of norovirus clinical samples. And now with actually only a limited number of samples, we are able to create these profiles. So how different is this data from what we observe in clinical samples? So what Rai did here is he compared the GenBank data together with the wastewater data in pink. And overall, we actually see quite similar diversity. But what was very striking is that, for example, G24, which really causes the majority of clinical samples, clinical infections, and is found most in clinical samples, was not that prevalent in wastewater. And I think that in part this might be due to a difference in pathogenicity, but we think that it might also have to do with stability or maybe differences in being attached to solids or being in the liquid fractions. But these things are important to further investigate. Also here, we made some phylogenetic trees. And for me as a norovirus researcher, this is really nice because for norovirus, there's not that much surveillance and often it's quite patchy. So either there's a lot of surveillance in the US and then at the other moment in Europe or in Asia. So it's always very difficult to compare the data. And what we saw when we plotted the GenBank data, which are the small circles, and the wastewater data, if we compare the diversity globally over a short time frame, we did observe quite clear different placements of the European wastewater samples and the Asian sequences. What it also it helped. So we also built a phylogeny for G13, which is the most prevalent G1 genotype. And again, the small circles are the GenBank data and the large circles are the wastewater data, and then the colors, the different continents. And sometimes it helped us to complement the data. So for example, based on GenBank data, you would assume that this lineage is only present in Asia, but with the wastewater data, we were able to show that it actually circulated globally. We also found many clusters like this. So these separate lineages, and then we detected it in a few countries in Africa and also in several years. So showing that there's really localized circulation, but we have not detected anywhere else. And this is something that we saw multiple times for noroviruses, but also for other viruses. And sometimes we detected what we expected. So for example, here we found Canadian or sequences in the Canadian wastewater that were identical to a large outbreak in that area in the same time periods. And also here, we saw many examples of these. So one step that we're now also taking is using these sequences from wastewater and using phenotypical assays to also investigate, for example, receptor binding and virus neutralization. So trying to really take this wastewater studies one step further. And this is work done by Putri Ayu Faryar, who's also a PhD student in my lab. So in conclusion, wastewater really contains numerous viruses, and many of them infect bacteria and plants. So if you are interested in monitoring of more clinically relevant viruses, it's really needed to use an enrichment method, either specific primers or capture-based methods. So if we looked at the virome, we didn't see clear continent level clustering, at the virus family or genus level. And maybe you would also not expect this, but for the bacteriome, they actually found very clear continent level clustering. We did see more clear differences in city level composition, virome composition. And what we also saw is that there were, we often found different virus types compared to clinical samples or a different distribution. And what exactly underlies this difference really requires further investigation. But I think for a large part, it will be either disease severity differences or shedding levels, or also the stability in these wastewater samples. But overall, I really think, just like a previous speaker, that global wastewater surveillance really has the potential to provide new insights in transmission patterns, but also in localized transmission of these viruses. And maybe the most important slide. So all this work was done by Nathalie Worp, Rais Guedalada, and of course, Marion Koopmans. And for the rest, I would like to thank our collaborator, Frank Aarstrup. And with that, I'm happy to take any questions.
Thank you so much for that super interesting presentation and joining us today. There are some questions in the Q&A, and I'd like to invite everybody else at the webinar to put any questions you have. I think any questions are welcome. There's no such thing as a dumb question. And I'll start with maybe the dumbest question of all, and then I'll get to the rest of the questions. But I guess, well, I don't want to call my question dumb, but other people can. I was curious about, like, you mentioned something about like the stability of the genomes and how that might affect your results. And I was wondering if you had any thoughts about, based on, I can't remember if you mentioned what kind of sequencing you used, but if you feel like the viral genomes, particularly for norovirus, since you took such a deep dive there, are like intact, and how do you feel like, were you able to piece together like metagenomes for norovirus from different sequences? And are you making, what kind of assumptions go into making like the phylogenetic tree? Like, are you working with metagenomes that you created from your samples? Or are you working with like one gene from the genome? Can you talk a little bit about that?
Yeah, so we used Illumina sequencing, and I think especially for this data set, the stability is important because we used frozen samples. So for the respiratory viruses, we don't freeze because you lose a lot. And for the, so we used quite long reads to assemble. I think making contigs is always a bit of a risky business from wastewater. So what we did is we looked at contigs with high coverage, and then also looked at how much variation you could observe for these contigs.
Mm-hmm.
But
do you
indeed,
feel like do you feel like the genomes are fragmented and that's affecting the interpretation of the sequences, or do you feel like the genomes are intact for norovirus? Like, what are your
I think so. The way we do the sequencing is that we pre-treat. So if virus particles are intact or not intact, the genome is not protected.
So you use like DNase or RNase? Is that
yeah,
okay?
Both, yeah.
Okay.
And so it's more from intact particles. So then you would have whole genomes. I think the more risky thing is that you probably have different viruses in one sample. So you still do combined sequencing of perhaps similar but slightly different viruses, because you have quite large capture, or you have quite large areas where the wastewater is coming from.
Okay.
So I think
I'll
that is a bigger risk.
Thank you. There's
okay.
So many questions here. So I'm not going to really go in order here. There's a question about the method, just while we're on the topic of the methods, and that someone asked about why you decided to go with the capture-based sequencing, because there's a concern that you would miss some of the emerging viruses that you don't have like probes for. And then there's also the concern that the viruses are always like mutating and evolving. And so maybe the probes you designed initially wouldn't work. Can you talk about that briefly?
So I think the capture probes, they allow for quite some differences in the genetic material. So indeed, a completely novel virus you would not detect, but novel variants of an existing virus you would certainly detect. So the other approach could of course be shotgun sequencing, but I think also to detect a completely novel virus there, you would need to sequence really to a very high depth, which is of course possible. But it would mean that probably you have a lot of plants and a lot of bacteriophages.
Okay, thank you. Speaking of the plants, there are two questions about plant viruses. One is asking about whether the tomato brown rugose fruit virus is common in European wastewater samples. Do you know of any recent measurements that suggest it's present? Because it seems to be quite high in the US wastewater.
I've not looked into how it currently is. So in the Netherlands, we're now doing another one of these studies, but I don't know how the situation is in Europe at the moment for this virus.
And then I think this other question I've heard come up several times, if you knew of any studies looking at the infectivity of plant viruses in wastewater, do you happen to know?
No, it's an interesting question, and I've also been contacted by some people from Wageningen University, which is the university here that studies plant viruses. We still have the samples, and to my understanding, these viruses are also extremely stable, so to assess their infectiousness.
Mm-hmm. There's a technical question about the PCA from someone who's interested in what dimensions you were reducing and how, like, whether you were using like read depth or read length or coverage as one of the variables in the PCA. So just like maybe a little bit more explanation of what the different dimensions are that were collapsed.
Ooh, so I'm not the best person to ask this one. But what we did is we adjusted the size of the genomes to start with. So we looked at the number of reads, but some of the viruses are very long, whereas, for example, norovirus is only 7,000 base pairs. So we adjusted that. And then we did a transformation to really compare the compositional data rather than absolute read counts, because otherwise you have too big of a bias between samples. But which dimensions were reduced, I don't know.
Okay. I'm guessing maybe if that figures in your paper, it probably has it really detailed in the method. So we can look there. Okay, so the last question we have, and I think we have time for just one more, so this is great, is related to human reads. In the US and at universities, we have to remove the human reads from our samples for various reasons, security reasons. But we were just curious, a question, one of the audience members wanted to know what proportion of the sequences that you pull out with your probe capture method are mapping to the human genome. Do you happen to know?
I don't know by heart. There are quite a lot, and we also have to remove.
Mm-hmm.
We've talked about analyzing the data because I know some studies where they did investigate that. I don't know exactly the proportion, but it is very interesting data.
And since we have one more minute, I guess I wanted to ask one more question, which is, have you thought about looking at any viruses with segmented genomes as part of, like, as in depth as norovirus? And do you have a plan for how to sort of put segments together
I think
bioinformatically?
well, I think segmented viruses are quite a challenge, especially with mixtures already. I think if you have co-infections, it's in one feces sample, it's already quite difficult to do that. So we have looked at some segmented viruses, but we've not tried to get the segments together. Yeah. I
All
think
right.
there will, yeah.
Yeah, it seems like a challenge that a lot of people will be working on. Thank you so much for sharing your amazing data set with us. It seems like you could be busy with that for the next 10 years, maybe, and continue to publish neat papers. Thank you, everybody, for attending, and we'll see you all in a month. Thank you. Bye.
Bye-bye.